So today we are here to talk about work that we've been doing for the last 2 years or so on improving HIV incidence estimation through integration of molecular data with epidemic modeling. That's a mouthful and it contains some technical terms, so I'll take you through what I mean by sort of epidemic modeling, et cetera. And what I love about this work is that it's highly interdisciplinary. And we really needed a group of experts to come together for the methods development that we've been working on. And so this talk actually will be by myself. I'm Natasha Martin, a professor here at UC San Diego in the Division of Infectious Diseases and Global Public Health. I'm joined by Assistant Professor Ravi Goyal and Assistant Professor Neera Mushiri, and each of us have worked on different aspects of this project. And so we'll be presenting kind of the different aspects that we have worked on today. And really brought together different types of expertise to address these problems. I also wanted to start by acknowledging that this work is done in collaboration with a huge team of researchers. We have, this work is really led by Susan Little. We've had input from a large number of statisticians that have helped us wrangle the data, people with experts in molecular epidemiology that have helped us interpret that, and statistical expertise, et cetera. So we've had a large team that you can see here on this slide, as well as the health departments that we're working with to make sure that the modeling that we're doing actually has public health and real world implications at the end of the day, which is the goal. Okay, so today we're talking about ending the HIV epidemic. This is the goal to reduce the number of new HIV infections by 90% by 2030. And in order to achieve that goal, settings such as county health departments and states need a good way of estimating the number of new HIV infections, since that is the key outcome metric that is the target, as well as a good way to predict the number of new HIV infections in the future, such as in 2030, and to use that information to understand how to optimally allocate HIV prevention resources. And so, the general idea is that county health departments and policymakers need to understand where they can make best use of their HIV prevention and treatment resources across different groups, geography, localities, in order to reach this incidence target in the most efficient way possible. So our team has expertise in developing epidemic simulation models to help answer some of these questions. And epidemic models are a type of model that mechanistically simulate infectious disease transmission between populations. And they can be used for a number of applications, but in particular, they're useful to estimate quantities which are hard to measure in the real world. So for example, true HIV incidence, which is hard to measure. They're used for prediction, of course, predicting the expected or number of new infections we might see next year or in 10 years, and look at predictions under different levels of intervention and scenarios of different interventions. And they can also be used, therefore, to inform understanding of how to optimally allocate resources. So thinking about the different prevention interventions we have for HIV and how they might be best allocated towards specific groups, either by, for example, race, ethnicity, transmission risk, or geography in order to reach our targets. And so there are a number of challenges in terms of predicting HIV incidence in the future. As I mentioned, it's very difficult to measure true HIV incidence in the population, in part due to delays to diagnoses, as well as differential testing patterns between groups and testing patterns that change over time. And those things make it hard to even really have a good handle of how many new infections we have today. And then, of course, that makes it hard to predict how many new infections to expect in the future. And the prediction models that we have to estimate HIV transmission in the future are often hampered by a lack of robust data in a number of domains. But today I'll be talking about, for example, aspects such as mixing between populations, so sexual mixing patterns, or for example, injection drug use mixing patterns by risk group, race, ethnicity, geography, et cetera. And so what this means is that these models, although they are useful in predicting into the future, if we don't have a good understanding of mixing between these groups, then the predictions that they make in terms of intervention targeting or resource allocation may be incorrect. So we may be recommending sort of the wrong options because we are lacking key data to inform some of the model parameterization. So traditionally, these epidemic transmission models are informed by data. We have epidemiological data. So this is data such as new diagnoses, HIV prevalence from sero surveys or cross-sectional studies or longitudinal studies, estimates of ART coverage, et cetera, sort of standard epidemiological data that are usually collected and reported by health departments. They're also informed where possible by behavioral data. For example, self-reported sexual and injecting risk in terms of sexual partners, injecting partners, etc. So those are two types of data that are commonly used to sort of either inform the parameterization or the calibration of these epidemic models. But there's an additional data type that could be incredibly useful, and that's molecular data. So data that we have about the viral genetic network. And that information, such as in phylogenetic trees that we can reconstruct from the genetic data, or characteristics of growing clusters or viral genetic linkages between groups, all of that kind of data potentially provides us very valuable information across a number of domains, but in particular, sort of transmission patterns, thinking about transmission between groups over time, etc. And so what you can see here on the left-hand side is the observed San Diego molecular viral genetic network. And you can see here that there's information here in terms of linkages and clusters that could be useful when we're actually trying to simulate these transmission patterns. So ours is not the first group to recognize that molecular data may have some value in informing our epidemic models. But there really has been a limited number of groups that have studied incorporating molecular data into epidemic modeling. And it's actually happened relatively recently, sort of over the last 10 years or so. The key challenges that have hampered a lot of growth in this field are methods development. So a lot of it, we needed to develop new methods in order to run these simulation studies and construct these and construct both transmission networks and molecular evolution and do this computationally, but also the very, very high computational burden that it takes to kind of run these simulation studies that really were, it was computationally prohibitive until relatively recently. And then a key issue that we have been able to overcome with the data that we have is about the observed molecular sequence networks that we see. Those being incomplete, they're only partial networks of the whole transmission network because not everybody is sequenced, but also who is sequenced is not random. And so we have this non-random sequence sampling that means that it poses challenges in terms of drawing inference about the broader network. And this is actually where we spent a lot of time, and Robbie in particular spent a lot of time thinking about how to address this. Okay, so our key question in this project was, can integration of molecular data improve our model estimates of HIV incidence to inform HIV prevention efforts? And we had three key challenges that we had to overcome in just even developing the methods and the methodological framework for addressing this question. And so each one of us will talk about one of these challenges and how we've managed to overcome it. So the first challenge was just methodological in terms of how we do this simulation modeling. So the simulation of a contact network, simulation of transmission on that contact network, and then simulation and then generation of the phylogenetic trees from that transmission network. And so that was one key challenge. The second one was, as I mentioned, we have this issue of this partially observed viral genetic network and the non-random sequence sampling. So we needed to develop methods to handle those issues, to ensure that we were sort of comparing apples to apples when we were trying to compare our simulation studies to data. And then finally, I've given you a few examples about how molecular data could be informative to models: phylogenetic trees, cluster growth characteristics, viral genetic linkages, viral genetic linkages, et cetera. And so one key challenge was just identifying what are the right calibration metrics from the molecular data that we should be using to inform our modeling. What are the ones that are the most informative to understanding transmission patterns and help us predict that in the future? So these are three of the challenges that we've tackled. And I'll hand it over to Nima, who will talk about how some of his development for the first challenge.
Yeah, thanks folks for joining us. So I'll be focusing primarily on challenge one. So just to give a little bit of context behind how we're simulating these networks, we start by having a joint simulation of the contact network and the transmission network. It's a joint simulation because the contact network is actually able to change over time. This is all very amazing work that Robbie developed in terms of how do we model these dynamic links between folks in the contact network. And then as those links are changing, how do we simulate transmissions across this changing network? And then the output of that is a contact network as well as a transmission network. And just to give a little bit more details about the contact and transmission network, we model three different linkage networks. We have a network of MSM sexual network, a heterosexual network, and an injecting drug use network. I want to emphasize that these are not independent networks. Individuals can span across multiple of them. So you can have, for example, MSM folks who have heterosexual partnerships as well, and MSM folks who have partnerships with injecting drug use. So you can kind of have individuals across multiple networks. But these are the three main structures that we're calibrating to. And yeah, individuals can either kind of be in stage one where they're HIV negative, or they transition to stage two where they're HIV positive, but they could be diagnosed versus undiagnosed, treated versus untreated, virally suppressed versus unvirally suppressed, or virally unsuppressed. So we sample a contact network and transmission network along that model, and kind of here's some more overview of the different kind of properties of this model. We have properties about the population, the transmission patterns, the health stages folks can be in, the care stages that folks can be in. All of this is informed by real data that we have collected. And then once we simulate the contact network and transmission network along this framework, we then feed the transmission network to a separate tool to then simulate viral evolution along the transmission network. So when person A transmits to person B, this pattern kind of constrains the evolutionary history that can exist within their bodies. Each of these transition events is kind of a branching point that is the latest point at which two viral lineages inside of the host can split. So there's a lot of constraints that it imposes on the phylogeny that you can receive. So under these kind of restrictions in mind, we simulate the viral phylogeny for each individual transmission chain of the transmission network, and then we merge those to create a global phylogeny across the entire population. And then the last thing is, well, you get a phylogeny out of this. Is it realistic? Is it not realistic? Who knows? So we then take the ground truth real phylogeny that we have from San Diego or from whatever population you're studying, and then we use the various metrics that Natasha mentioned to compare how realistic the phylogeny we get at the end of the simulation actually looks against the real world. So our goal here, we want to be able to calibrate parameters in the contact network and transmission network stages. And we want to be able to determine what metrics should we look at in the downstream molecular data. So first, I want to give an overview of how we initialize the phylogeny of the viruses. So there's a framework that I developed called Phavites, where you can basically simulate viral phylogenies and the contact network and the transmission network under various models. And in the original implementation of this that we had presented at a previous conference or previous presentation a while back, the trees that we got, they did look realistic, but unfortunately there were certain metrics that weren't actually as consistent with the real data. So just kind of looking at it broadly, it had some properties that we did expect, but some of those kind of concrete metrics weren't matching up as well as we wanted. So we need to ensure that the seed tree, basically the viral phylogeny that's ancestral to the epidemic, so basically up until 2019, the start of our epidemic simulation, we needed to ensure that that tree was consistent with the real tree that we had up until 2019. So our solution was basically each individual person in the simulation is mapped to a real person in the population from whom we have data and from whom we collected data to build the real phylogeny. And then rather than trying to simulate a brand new phylogeny to kind of seed the epidemic, we just take the real existing phylogeny and map each person from the simulation to somewhere in that tree. And then we kind of use that as the basis. So what does that look like? We basically, oh, it's kind of cut off, but let's see if I can hide the controls. There we go, perfect. Okay, so we start with the real phylogeny that contains all the actual individuals in our population that we have sequences from. And then, whoops, did I break something? Oh, there we go. And then the epidemic simulation tool, it'll simulate a phylogeny alongside each individual transmission chain. So basically, each individual that was infected at, that was HIV positive at time 0, seeds some transmission chain, and then we simulate a phylogeny along each one of them. And then for each simulated phylogeny from each transmission chain, we find where exactly it should be placed on the real seed tree. So for individuals who do have a counterpart in the real tree, we'll map it somewhere proximal to that person. For individuals that exist in our simulation, but from whom we don't have sequences, we map them somewhere based on their actual demographic properties. So we take advantage of the fact that we know all the demographic details about the individuals in the real data set. We have demographic properties about every single person in the simulation, and we try to place people with some probability close to where is kind of densely comprised of that demographic breakdown. You'll notice that these attachments are kind of differing lengths. So we have a phylogenetic model to actually randomly sample according to a Yule process where exactly these branches should kind of be merged into the real tree. And then those parameters are informed from real data. So just looking at a few calibration properties that previously were not matching up with the real data set, now we do actually get much better matching up. So on the left, we have the actual observed tree where we remove anyone that was sampled after 2019. And then we have the simulated seed tree, so the tree that we kind of start seeding the epidemic simulation. And you'll notice that, and I don't want to get into the nitty gritty of what these metrics mean, but basically across various metrics of tree balance, as well as of kind of tree branch length distribution, we seem to be lining up much better now. So in summation, with regard to challenge 1, we have now come up with a framework that is able to recapitulate a lot of the properties we were hoping for in the real genetic data. So maybe I'll hand it off to Robbie for our second challenge that we tackled.
Thank you, Natasha. Thank you, Nima, for going through those. And then I'm going to be talking about the second challenge, which is how do we deal with that we're only observing a partially observed viral genetic network and as well as non-random sequence sampling. So just to get a little bit of idea what that means and sort of walking through an illustration of that. Here we have a sort of a depiction, and the circles here are going to represent individuals who have a sequence, and the links between the circles are indicating that their viral genetic sequence between the two individuals are similar at some kind of threshold, and so that defines a link. So here in this picture, you can sort of think about we have a total of 7 individuals, where some of them are linked to each other. Well, of course, there is going to be individuals who are also part of this transmission network overall. We're not going to be sequenced, so we're not going to be able to see who they're genetically sequenced from. And even further, there's also going to be individuals who are undiagnosed, so clearly we're not going to have any information on them in terms of their sequence. So if we were able to observe the entire picture, if we had no missing data, this is maybe what the complete phylogenetic network may look like. But clearly, that is not what we're observing from the data. And for San Diego in particular, we see that about 47% of individuals have a sequence. And so to address, so obviously we have to address this problem. We can't compare, you know, our simulated or using the framework that Nima just went over and compare it to something that has a lot of missing data, where we can't do apples to oranges right there. So what we do is we sort of thought about two different approaches to tackle this issue of missing data. The first one, so I'll go through both of them. The first one is a kind of a mechanistic approach. And so we're going to go very much leveraging the framework that Nima went through and walk through how we can start with that simulation and figure out how we can sort of think about the mechanisms that lead from the complete network to that partially observed network. And so that's going to, and I just want to walk through a little bit of an example on how that works. And so first, through the simulation, what we can have is we'll have everyone. We'll know who is in the simulation. Everyone is in there, and we'll be able to sort of think about what their sequences are, who's diagnosed, etc. We'll have that kind of information. But when we want to compare it to the true thing to help calibrate it, we're going to sort of start walking through the different mechanisms that lead to the partially observed network. And so the first one that we do, we have a mechanism that sort of Nima talked about when they go through the different care stages in an individual. Some of them will be undiagnosed and some won't be diagnosed. So that we can mark and we can remove those from our simulated data, those who are identified as undiagnosed. And then the second step is we can think about who gets sampled and who doesn't. So we're able to then look at the actual data that we have from San Diego and estimate what is the probability based on various covariates, characteristics of the individual, whether it's their race, ethnicity, whether it's their transmission risk characteristics, and we're able to sort of assess who and what probability that individuals are going to be able to, who are going to be sampled. And so we can start mimicking that mechanistic process in our simulation that Nima went over. And so by the end, in our simulated epidemic, we'll be able to recapitulate, hopefully, what looks very similar to the observed network through these different mechanisms of being diagnosed, being sampled, having their sequence sampled, and et cetera. And then hopefully what we simulate here will look very similar to the observed network. That's the goal, and that's part of the calibration process that Natasha will go through a little bit on challenge number 3. But then there's also, so this is what that first approach is very much understanding the mechanism that and very carefully delineating that. And that takes a lot of data and a lot of assumptions to sort of think through. The other approach that we can sort of do, and that we're referring to as sort of statistical inference, is taking what we have in terms of data and trying to go backwards and impute upwards. And so the idea is to estimate the full molecular network or viral genetic network from our observed sequences. And so here we have developed a Bayesian inference approach to be able to take our partially observed network and impute or make inference about the complete network. And this approach leverages a lot of work from some of the collaborators there on developing a very flexible network model. So we're going to leverage that and that prior work that we've done, and as well as recent advances around graph enumeration. So those kind of two things allow us to really be able to computationally handle being able to do this imputation in a Bayesian setting with larger networks. And so before this work had limitations around the computational ability to be able to do this. And I'll sort of say one thing about this approach is, once again, we have this very non-uniform sampling, and this statistical inference approach puts kind of limited restrictions on exactly how people are sampled to become sequencers. We're just going to take it as observed. And so what's really nice is that we can scale this up with some kind of computational efficiencies that we have here and on our approach, and being able to actually do this inference on the size networks or that is actually observed in San Diego, in Miami, and other places. So just now I want to walk through a little bit some preliminary findings that we have seen from using the statistical inference approach. And so here, but first I want to give a little bit of a background from Miami-Dade County. And in particular, just giving a little bit of context on how much missing there is and the differential mixing by different characteristics. So here, this information is only for MSM, so it's kind of a very restricted set. But here we can sort of see what we have here is different race characteristics and different transmission risk, and the number of individuals who have a sequence and the number of individuals who don't have a sequence. And then in particular, is what is maybe of importance is the last column that gives what is the portion of people by characteristic that have a sequence. And we can sort of see this varies from 38 for race, from 38% down to 21, 22%. So we actually do see very much differential sequence sampling. And so what we did to sort of assess how well our Bayesian inference approach is, we did a simulation study, because obviously the only way you can assess it is if you have the whole truth, and our data doesn't allow us to have the whole truth. So what we did is we simulated a truth. We simulated what a network, a viral genetic network would look like. We said, okay, this is the ground truth. And then we implemented sort of this sampling process of maybe only observing about 30% or of the individuals would have a sequence, which is roughly, we see 30, 31% in Miami-Dade County. So this is what the observed network would look like. And I should sort of say, what is the network property that we're doing here? We're just doing it by degree. And degree is the number of linkages that an individual has. So here, for the observed one, about 13% would have 0 linkages. That's degree 0. And a little shy of that would have 1 linkage coming out of them, and so on. And this one, if you, on the observed network or on the partially observed network, we would have a high number of people who would be unlinked to anyone else. And so when you're only seeing 30% of the data, you're only seeing about 9% of the linkages. And so you actually are going to have this huge number of people who seem unlinked. And so in this simulation, when you do this sort of subsampling, you would have about 80% of people would look like they're unlinked, even though in the real data it's about 13. And when we back it out, we get about an estimated 21. In our procedure, you can sort of see how well we kind of do. And we match it, not perfectly, but much better than just using the raw data. And so we're getting pretty, I would say, not too far off. And it gets better as you sort of sample more and more individuals. But want to show an example about the samples coverage that we're seeing in Miami. And so you can apply this not only to degree, but going back to mixing patterns, which is one of the key things that we wanted to kind of look at and understand. So here is the true data that we observe in terms of linkages across different race and ethnicities. And I'll just sort of go only through the first bar, but then all the other bars are exactly the same. I'll go through the first data point. So this bar represents Black individuals, and it gives you the proportion of linkages that Black people living with AIDS, HIV have with other race and ethnicity communities. So, for example, you'll see here that linkages for Black MSM is about 58% would also go back to Black MSM. So Black MSM would be linked to another Black MSM. About 37% would go to a white MSM, 5% to, 37 to Hispanic, 5% to white, and about 1% to all the other races. And that is what's observed. But when we do our imputation process, we see a very different mixing pattern there. And that is partly driven by the differential rates that blacks and whites and Hispanics are being sequenced. And so we can see quite a big difference in that. And so that's going to have probably a big impact on our understanding of the transmission dynamics. And so just to sort of conclude where we are on that, so we have this big challenge of missing data. But we sort of developed two different approaches that allow us to tackle this. One is more mechanistic and one is statistical, and I think they both have their pros and cons on helping us address the overall goal of the project.
Great. Thanks, Ravi. Thanks, Nima. All right, so it's left to me then to describe where we haven't quite solved the problem and where we have ongoing work. And that is in this third challenge, which is the choice of calibration metrics from the molecular data. Nima already talked a little bit about that when he was talking about the tree metrics, the balance metrics you were talking about. But we have, this is a sort of an ongoing area of exploration that we're still working through. There are really no established methods for what features of the molecular data are informative for model calibration. And it may be that, models that have different goals in terms of estimation of transmission between risk groups or understanding current transmission may have different metrics that are more useful for calibration. So I don't think this is necessarily, a one-size-fits-all problem, and we need to kind of understand what is the best data, best metrics for our question. So we had initial discussions that you've already heard about wanting to ensure that at least at initialization, our tree metrics looked right. But then we also really wanted to think about these linkages between groups, just like Ravi was talking about. There's information in what we see in terms of who is linked to who. And so as a first try, what we decided to do was start with these metrics of the proportion of genetic linkages by transmission risk group. So this is, Ravi was just showing work that he's done in Miami-Dade. I'm now going to take you back to work that we were doing answering similar questions in San Diego County. But we were really, so we wanted to look at, for San Diego County, we were very interested in looking at transmission by risk group, which is why we chose this particular feature to look at. And again, this is sort of something that's a standard measure in molecular studies. This is Joel Wertheim's studies looking at genetic linkages by race ethnicity in a few different counties where they look at these proportion of links and look at how that varies between groups. So we did the same thing, but there are, for our analysis, and I'll show you those results in a moment, but there are other potential metrics that we could have chosen. And I wanted to kind of just throw them out here so that we could all think about what might be more useful. So I just showed you information that we used for our current simulation study about linkages between groups, but you could also look at cluster characteristics. And in particular, we might be very interested in growing clusters and where we see clusters of individuals who are linked together, but that there are new individuals being added to those in the last year or 2 years, active of a sort of growing or active HIV cluster. And so, for example, in San Diego, we've been able to characterize the growing clusters and the stagnant clusters and their characteristics. And we observe that growing clusters are more likely to contain heterosexual individuals and less likely to contain monosex men. So that might be something that you could use for calibration, especially in understanding kind of current incidence patterns. We could also look at cluster growth by geography. So this is looking at clusters, and the size of the dot is the cluster size. The color of the dot is whether it's a the speed of the cluster growth, blue being fast growing clusters, orange being slow, and red being stagnant. So you might be interested in saying, okay, well, maybe we want to calibrate, if we have a model that incorporates geography, maybe we want to incorporate information on these growing clusters and their sizes by geographical region. We could also use information from phylogenetic analyses. So this is work that was led by Antoine Cheyenne here at UCSD and colleagues, where they were looking at the San Diego-Tijuana border region and built phylogenetic models to understand spread and dispersal. So who's acting as a source and a sink for HIV putative transmission events. So here, for example, you can see that there are arrows that are telling us basically where we see the directionality of HIV transmission between risk groups. So MSM to PWID MSM is in green, MSM to MSM is in yellow, heterosexual to heterosexual is in blue. And so you can start seeing these directionality in terms of transmission cluster events between, for example, even San Diego and Tijuana. So incorporating geography as well as risk. And we can use the same phylogenetic information to also understand the sort of relative contribution of the migration events and risk groups to transmission. So of transmission events where we see migration events, so from one group to another or one geographical area to another, what are the sources of these events and what are the sinks of these transmission events, either by geography, which is shown here on the left, or risk group, which is shown here on the right. So for example, if we just look on the right-hand side, we see that a large fraction of transmission events from one group to another occur from people who inject drugs who are heterosexual to MSM, which is a really interesting feature that, again, we might want to incorporate into our model. Okay, so this challenge is still sort of in process. We haven't solved it, but the benefit of this sort of flexible framework that we've developed is that we're able to test this and sort of incorporate these different calibration metrics and see how it affects our simulations in terms of HIV incidence in the future. So this is ongoing work. But we would like to show you some very preliminary results of simulations that we've done, just incorporating the proportion of linkages calibration metric that I mentioned. And we applied this model to San Diego County. So our question was really, well, if we additionally incorporate this molecular data into our modeling process, does it change our result? Is it useful? Is it of increased value to our simulations? So what we wanted to do was we wanted to compare. We simulate HIV transmission in San Diego County. And for one model, we calibrated only to the epidemiological data in terms of new diagnoses, ART coverage, etc. And then we simulated another HIV transmission model using the framework you've heard about. But this time we calibrated to both epidemiological data as well as the genetic data. And so, for example, new diagnoses and the proportion of viral genetic linkages by transmission risk group. So we have these two simulation models. They're both calibrated to the observed epidemiological data, but one of them is additionally calibrated to the genetic data. And we take those two simulations, and for each one of them, we assess how well do they fit the data, and what do they project in terms of future incidents, and as in particular, what do they project in terms of future incidents if we have intervention scale up? Do we see differences in these interventions? Do we see differences in incidents? And so in this talk, the intervention that we'll show you is just simulating testing scale up among one group, MSM, but you know, you could do this process. That's just illustrative of whether we see differences when we simulate a single intervention. All right, so these are very preliminary results. They literally only finished running this morning, but I'm really excited to show you them. So remember, we have these two models that we've calibrated. Both were calibrated to the observed epidemiological data. That's what we call our SIM-E in red. One is calibrated to our epidemiological and genetic data. That's our SIM-E plus G in green. And the observed data here is in new diagnoses, and that's in blue. So we have a couple of years of observed data of new diagnoses. Here, I'm showing you new diagnoses over time, and each panel is a risk group. We have people who inject drugs on the top left, MSM who also inject drugs on the bottom left, MSM who do not inject drugs on the top right, and other individuals on the bottom right. And so what we're doing here is we're looking at the observed data in blue, and we're trying to see whether our simulations match these observed data. And there's quite a bit of, there's a little bit of noise in these data points. We see some variation year on year in new diagnoses. And there's uncertainty in the model projections, but broadly speaking, the projections for both the simulated epi only and the simulated epigenetic calibration simulations matched the sort of historical observed data in terms of new diagnoses, and they're pretty similar in terms of their predictions between these. And we kind of would expect that, right? They were both calibrated to the observed epidemiological. But where we start seeing very big differences is when we then take those two simulations and we say, well, how well do they match the observed molecular linkage data? And so for what I'm showing you here, I know this is kind of hard to read, but basically, again, we were calibrating to those stacked bar charts that you saw in terms of the proportion of linkages by group. If we take each one of those proportions and now they're an individual bar on this this very long bar chart, what we can do is the blue is the observed data, so that's the observed number of linkages that we see, and then the orange are the predictions from our simulations, and on the left hand side that's our epi data calibration in orange, and the right hand side the black is our epi and genetic data calibration. So what you can see here on the left-hand side, just comparing our model that's only calibrated to the epi data, unsurprisingly, it doesn't match very well in terms of its predictions of the genetic data, which we would expect. It wasn't calibrated to that data, right? So we can actually simulate a sort of infinite number of networks that would, for each one of them, we'd be able to calibrate to the data in terms of our new diagnoses, but it wouldn't necessarily match our epi data. And so we see a big mismatches here between some of the groups. On the right-hand side, though, now you can see we have a much better match to the data. We have directly calibrated to this data, so we were expecting and anticipating to see that this model would have a better fit to the data. So of course, we don't know what the ground truth is. We don't know the true HIV incidence. We can't validate these models, but we can say that a model that incorporates both the genetic data and the epi data for calibration does a better job of recapitulating all of the observed data that we see here, in particular, of course, recapitulating the genetic data as well. And so that calibration process gave us information about the parameters that we're using for this model. And what we found through this epi and genetic data calibration is that it indicated that there was higher transmission risk due to injection drug use than in this model where we weren't calibrating to the genetic data. And there was also less preferential sexual mixing by transmission risk group. So for example, MSM there was less preferential choice of a sexual partner by, for example, ITU status among risk groups. And so these sort of differences that we then were able to infer by additionally calibrating to the genetic data, we wanted to know, well, does that have implications in terms of our model's projections in the future? So first, we just looked at what are the model projections of the true number of new HIV infections. So this is true new infections. Before I showed you new diagnoses compared to this data. This is now true HIV infections without intervention scale up. And without any intervention scale up in the next 10 years, the model is predicting that new infections would just remain stable among each of the different risk groups in both models. We would expect that. If we're not scaling up interventions, we wouldn't expect any change in epidemic dynamics. But what if we increase the intervention among a particular risk group? So for example, here we're just simulating a theoretical intervention of increasing the diagnosis rate among men-of-sex men in the top right by 5-fold 2024 onwards. So you can see that with no intervention, it was sort of the incidence among MSM was stable or just slightly rising over that period with both of our models. And now with this intervention of increased diagnoses, that means that more MSM are diagnosed, more are initiated on ART. We see a reduction in new HIV infections. Now, this is a big increase in diagnosis rate and sort of a relatively small increase in new infections among MSM, and a lot of that is because of this process by which people are initiated on ART, drop out, lost to follow-up, etc., which is hampering the benefits of new diagnosis. But okay, so both models predict this sort of decline among MSM, but they have differential impact among the other risk groups. And it's a little hard to see in this figure. So what I'm showing you here now is if we look at the relative reduction in incidence among a risk group with the intervention compared to no intervention, then we start seeing differences between our simulations. So on the left-hand side, it shows that with our simulated intervention of increased diagnoses among MSM, both the simulation which is epi data only, and this, which is in the red, and the simulation with our epi and genetic data in the blue or blue-green, both simulations predicted a similar reduction in incidence among menopausive men with that intervention. But we start seeing differences in predicted impact of interventions among other risk groups. So for example, we saw a 27% lower impact of that intervention predicted among menopausive men who inject drugs with our epi and genetic simulation. And that was because of those two factors that I told you about, that there was this sort of simulation inferred that there was more injection drug use related transmission, and there was less preferential mixing. And so we see different impacts on the broader network because our network is different in these different simulations. And we changed our network and informed our network by using the genetic data. So I think this was really a proof of principle that we could do this process, and that if we additionally just included one method of calibration, which was our proportion of linkages, it could both, we could calibrate to it, and it could generate differences in terms of predictions of incidence among risk group with particular interventions. And of course, if we're seeing differences in our intervention impact among risk groups based on incorporating the genetic data, then we would anticipate that we might see differences in terms of recommendations of resource allocation to risk groups based on this updated and more informed model. All right, so in conclusion, I'm really excited to be working with such an awesome team that's developed this novel methodological framework to incorporate molecular data into epidemic modeling. It's still work in progress. We're still refining sort of the model calibration. It's very computationally intensive, and so we actually still need more time to kind of do all these simulations. So what you've seen is just, again, preliminary work. And we know that there is intervention change over time. So for example, PrEP is being scaled up on MSM in San Diego County. And so we want to incorporate that into the model and then use that to simulate what we anticipate incidence to be in the future based on that. There are other molecular features which may be informative. I went through a few of them. We will be exploring those. And in particular for our Miami project, we're very interested in the geographical heterogeneity of HIV in that area. So we'll be extending the model to incorporate transmission in both by race, ethnicity, transmission risk, as well as geography. And that will allow us to then additionally calibrate to some additional information that we have in terms of that geographical heterogeneity, which is future work. And ideally, we will be able to validate the models at some point, right? We don't know what the ground truth is, but we can use the models to make prediction, and then we can hopefully collect data in terms of new diagnoses in different risk groups, and ideally in the future actually validate the model predictions. And so, as I mentioned, this work started in San Diego as part of the Triumph project. We will be extending it over the next few years to Miami-Dade County. That's a project that's funded by NIMH. It's led by Susan and myself. And ultimately, what I've shown you today was a lot of transmission modeling. But ultimately, we're going to bundle that into a simulation model that simulates all of this, plus HIV resource allocation. So we're counting the dollars about how we're actually doing these prevention, interventions, testing, PrEP, et cetera, and to actually provide information to the health department in terms of how they can better spend their HIV prevention resource dollars, again, by group, such as race, ethnicity, or geography, to maximize the benefits and improve health. So I'm really excited to be working on that. And a big thank you again to all our collaborators. the funders, and just to sort of reiterate that this has been a very big project, but also a highly collaborative project also for the San Diego CFAR. We're here representing the Biostatistical Modeling Corps. We've been working with the TV Corps. The HAS Corps is part of this project, and so we're really working together to solve this problem. So thank you very much, and I'm happy to take any questions if there are any from people here or on Zoom.